We have today Dr. Stephen Hyman. He's a professor of stem cell and regenerative biology in Harvard University, and also the director of Stanley Center for Broad Institute. Thank you for being with us today.
Pleasure.
Thank you for your time. And could you briefly talk about your presentation? I think that's tomorrow.
Yeah.
Right.
Yes.
Titled "A New Molecular Map of Psychiatric Disease Mechanisms."
So psychiatry, as I think people know, is attempting to treat, hopefully one day prevent, a number of very severe illnesses that affect populations worldwide, most commonly the mood disorders and anxiety disorders, but also very severe disorders like depression, autistic symptoms, schizophrenia, bipolar disorder. And the talk is really focused on new developments that allow us, we hope, to understand the mechanisms of the disease, that is, at a molecular level, what's going wrong, so that we can ultimately design new biomarkers to stratify our patients and then, of course, come up with new, safe, and effective treatments. To date, we have a lot of very useful drugs, antipsychotic drugs, antidepressant drugs, lithium. But all of these drugs, even the newest ones, ketamine was just approved in the United States, and there's a new drug for postpartum depression called brexanolone, but all of them were based on human observation. So in essence, a prepared mind observing what happens when a human being gets the drug, but with no knowledge of mechanism. And the reason that we now know something about mechanism, which is what I'm going to talk about, begins with the revolution in genetics. And then I will talk about how we're trying to turn this rapidly emerging genetic information. I'm going to focus on schizophrenia, which is the most advanced, but it's relevant to all these diseases. How this emerging genetic information is giving us insights into mechanisms and, you know, the first forays into genetically driven biomarkers, research, and ultimately therapeutics.
Okay, thank you. And you were the director of National Institute of Mental Health in late 90s.
Yes.
And looking back and, compare today's recent trend, do you have anything to share with us in terms of changes or direction?
Well, the changes are enormous. I mean, psychiatry has dealt with very difficult problems. We have no direct access to living human brain tissue. So it's very much unlike cancer research, where a scientist can stand outside the operating room and a surgeon does a biopsy and hands you the disease. We rarely get human brain tissue, and we essentially never get relevant human brain tissue. And what's more, because there were no objective diagnostic criteria, all of the diseases have been identified by fairly arbitrary criteria, the DSM, the ICD, which creates a lot of noise. And we don't have very good animal models. We certainly need animals to study biological mechanisms, but we don't have animals that have schizophrenia. In fact, we don't have animals that really have Alzheimer's disease. I mean, the limitations of animal models for human brain disorders is another real challenge. So what's happened is that in the last decade, the technologies that came out of the Human Brain Project and very powerful computer tools, including both data storage, but also algorithms, have permitted us to begin to understand the causal genetic underpinnings of schizophrenia, bipolar disorder. They're the first forays in major depression, even anorexia nervosa. And these studies are unbiased. That is, they're not dependent on biological hypotheses. So we're learning things we never expected, and certainly looking back to when I was NIMH director, we couldn't have imagined. So these new technologies give us insight into causation, and then putting them to work requires, the use of animals to study mechanisms, but ultimately it requires human biology, right? And another remarkable advance is the ability to make stem cell models of human neurons. And of course, this was a recent and remarkable product of Yamanaka in Japan, who figured out how to make stem cells out of adult cells. And now many groups around the world are making different kinds of neurons and glia and even brain organoids, which we think will prove useful. And then there are technologies for genome engineering. I mean, CRISPR was certainly not imagined back in the 1990s, which help us engineer these models. And at least for monogenic, rare monogenic neurodevelopmental disorders might one day be used therapeutically, although it's still not quite ready for prime time. And then finally, there are remarkable technologies like optogenetics and in vivo microscopy that are permitting us to understand systems-level neurobiology. And so when I left NIMH at the end of the last century, I'm old, right? There was really very little traction on disease mechanisms. And as director, I focused resources on building up the necessary neurobiology, the kind of computational approaches to genetics that we imagined we would need, and we did need, began collecting DNA samples and phenotypes, hoping that the technology would arrive, and ultimately it did. And by the way, the samples were much too small because we really guessed wrong about how complex these things would be. And now we're living in a very, very different world.
Right, right. Thank you. I was going to ask you actually, ask for your advice on Korean neuroscience community, because Korean neuroscience community is now trying to move toward somehow collaboration between lab research and clinical research to let the lab knowledge and technology get into the normal people. But I think your answer actually implied much of it. But.
Yeah, but I think it is important, but I think you have to be careful. There's a push toward translational research because the problems are so severe. We want to do something about autism. We want to do something, well, everybody should want it, right?
Yeah.
But if we do it prematurely, then we're just guessing and we waste resources. And that's really been the history of psychiatry. So these collaborations are very important, but they have to be at an earlier stage. People have to learn each other's language. And I think one of the really important places to focus is for at least more basic geneticists to be working with the translational and clinical community on well-phenotyped cohorts that are large enough. And by the way, this requires global collaboration, not just collaboration between basic scientists, right? But global collaboration, and that's really been happening in psychiatric genetics, and it's included Korean scientists. But it's the complexity of these diseases is such. The signal to noise is so unfavorable. We need enormous cohorts, and we also learn new things by comparing genetically different populations. So there's that. But there's also the development of human biology. I think what we've realized, I had mentioned before, that we can use laboratory animals, rodents, to study mechanisms, but we shouldn't think that they get these diseases. We can't ask a mouse if it's hearing voices, and mice in Alzheimer models get plaques and they get tangles, but they don't get Alzheimer's disease, right? Sometimes I joke sardonically, there's never been a better time to be a mouse with Alzheimer's disease. We can cure you, but it doesn't work for the humans.
I see.
And so working together between basic and translational sciences to advance various human cell models in a dish, whether it's being able to make new cell types for, out of iPS technologies, but importantly, of people with many, many different genotypes and different levels of disease risk, again, because of the complexity, or brain organoids, or working together to connect brain imaging, so high field strength MRI or resting state connectivity with cellular neurophysiology that's rarely will be in humans. Sometimes epilepsy patients will consent to be recorded from, for example, but will also require animal research, hopefully including primate research. And I think these kinds of, not guessing at a therapy, but rather these kinds of fundamental, build the toolkit that we need to advance from the laboratory into the clinic And I think that will be amply repaid. And I think it's really a very good thing that there's that Korean neuroscientists want to do that.
Thank you for your insightful remarks. And lastly, you see many young faces in this conference site.
That's good.
Any advice for young neuroscientists in the future?
Yeah, I think it's really important, first of all, to get a diversity of training experiences and not, only work with one mentor. And I think that's important because increasingly neuroscience, and especially if we're interested in diseases, is becoming interdisciplinary. And having the skills to integrate different kinds of information is really critical. The second thing is everybody's going to need more quantitative skills than was the case not very long ago. Again, the size of the data sets are really very, very large and complex. And even if, even for somebody who's going to be a hands-on wet lab researcher, the ability to understand quantitative data and compute on their own data is going to be really, really very important. And then I think one thing that the young people are doing much better than their mentors, but you got to be careful because, human nature being what it is, people want to hold on to their data. But I think this idea of within the bounds of protecting human privacy, sharing data sets, and allowing larger communities to work on these data sets is highly advantageous. And it's a very different habit than people had when I was coming along. When I was an IMH director, we had the worst time getting people to share disease-related data sets on autism, for example. And I used to say, well, we're a public taxpayer-funded grant-making institution. The public is giving us the money because they want us to solve the problem, and yet people would hold on to their data sets. Sometimes I think they wanted to be buried with their data as if they were data pharaohs. And I think the younger generation, again, I'm much more optimistic about this, but I think it's really important.
Okay. Well, Dr. Hyman, thank you for being with us.
Great pleasure.
I'm very honored.
Oh, no, it's fun.
I hope you'll enjoy the rest of your stay in Korea.
I hope so too.
Well, thank you very much.
Thanks.
Thank you.
Okay.